One of the recently published devices where we image animal from below. And it turns out that when we image animals from below, we see much more subtle changes in their behavior compared to when we image animal from above. So another advantage is that this is a so-called this black box device, is that it uses infrared camera. Also, in fact, animal is placed in the darkness, and so rats and mice, they are nocturnal animals. They feel safe in their completely dark environment. They think that nobody is observing them. And we can combine infrared images with also total internal reflection images, and which allows us to follow weight distribution between all different poles of the same rat. And you can also apply it both for mice and for rats. And then by using machine learning and computer image analysis, we can then, without any labeling of the animal as such, we can automatically extract positions of limbs and body posture, and then quantify this depending on whether now in our wild-type animals or in animals where pain has been induced. And so when we, for example, in this type of data, when we injected complete front adjuvant to one paw of the animal, we see the weight distribution is dramatically affected in that animal will avoid using injured leg. But what is most important, and this is of course not, so this is fully expected what we see, and this is very easy to, in fact, quantify also manually. But what is more important is that we can actually, by applying known painkiller, which we know that has effect also in humans, in this case this is tanezumab. This is a monoclonal antibody which is binding to neural growth factor, which has been in the clinical development for and showing efficacy in humans for osteoarthritis pain treatment. We see that by applying this tanezumab, we see that we can affect this pain response. We can rescue these animals, showing that not only we can see the phenotype, but we can also, by using known painkiller, affect this phenotype. And this is showing that, okay, well now if we apply our own compounds, we can discriminate between analgesic or non-analgesic effect of these compounds in this particular assay. Similarly, when we apply the monosodium urate, which is now osteoarthritis pain model, depending on the time in culture or after addition, we also see that we see nice development of this phenotype during time, which then can be rescued by tanezumab treatment. And this is now fully automated, and this is now can be done, and so these studies are in fact done by a master student in the lab, so you do not need to be highly trained, skilled research assistant in order to obtain this type of study, which is of course greatly simplifies our life as a pharmaceutical company. You can also analyze much more animals within a given time period, which allows us to get more robust, reproducible data, and also compare and cross-compare results from different studies and from even different studies done in different research, in different projects, but even in different years. And so now, since like several years we've been doing this, we see that this is actually pretty much consistent data we obtain. We extended now to more neuropathic pain models. And in this case, this is a spared nerve injury. And so this is now still work in progress. But you can see, so for example, this rat has been suffering from this injury. And you see the weight distribution on normal, non-injured, and injured paw, which is very, very seldomly used. So this type of data you can now get with this newly developed methods of automated and unbiased machine pain assessment. And we believe that this is actually the future of behavioral analysis. Moreover, why we think so? Because now we are talking only about, so I've been showing you only like one parameter, weight distribution. But in fact, this system can be used to assess much more than weight distribution. It can also assess gait, so changes in the way animal moves, but it also can assess the other parameters like grooming, scratching, being on the like hind, it's like vertical movement, and this is currently which we are trying to also validate for pain, for assessment of pain, like in a more holistic approach. So we believe that well, it will also allow us to use more than one parameter for description of behavior, so more holistic description of the whole animal behavior. And then you can do some kind of more complicated assays like multiparametric clustering. You start seeing the differences in behavior in response to compounds in different animal models and being more sensitive to assessment of subtle phenotypes, of subtle behavioral changes which you induce in these animals.